Go.
Alright, this is the explaining like the glycolysis, Krebs cycle, and all that. So to begin, you're here with glycolysis. So you have to start glucose, and you want to get to 2-pyruvate. So what you do is you go across this horizontal line, which costs 2 ATP, right? But once you reach this point right here, you gain 4. So gain 4, lose 2. That's why you gain with a net positive 2 ATP. And alternatively in this process, 2 NADH plus 2H plus is also gained. Glucose is C6H12O6. Pyruvate, 1 pyruvate is C3H4O3. But since you have 2 of them, it comes out as C6H8O6. And you're wondering where the 4 extra, or not the 4?
8.
Yeah, where the 8 extra, or the 8 other H's went. So right here, you have 2 NADH, 2 times H, or 2 times H1, that's 2. 2 times H, that's 2. 2 plus 2 is 4. And then down here, you have 2 times 2, that's also 4. 4 plus 4 is 8. So the 8 are in this little area right here.
Area right there. And then the part where Luke's talking about the 2, which is 4, is talking about the energy investment, energy payoffs. Because when you're adding that, when you're putting in the 2 ADP, it produces 4 ATP.
That's why it's called an investment. You're investing 2, you're gaining 4.
Yeah.
This all occurs in the cytosol, and every name you'll see will be in red. And then move on over here. A little side note: these dotted lines, as you can see, if oxygen isn't present, everything right of this in blue and red is all just void. None of it can occur, and your plant will die if it doesn't get help soon. But next up, there is oxygen. We're just going to assume there is because there should be. You have 2-pyruvate. You're taken over to the pyruvate oxidation stage, where you have a pyruvate that move across this line. No ATP, no nothing is required for this line, but the coenzyme A attaches on this line and that makes the pyruvate become acetyl-A. And as a byproduct and a product, the NADH is produced along with carbon dioxide, and then more carbon dioxide is produced here. So, but remember, there's 2 pyruvates, so this process, just imagine, occurs twice, which is why up here you have 2 acetyl coenzyme A, and this all occurs in the mitochondrial matrix. Now we'll follow this line all the way up here into the light-independent section, which is the citric acids, also known as the Krebs cycle. They're synonymous. This one turns the acetyl coenzyme A into citrate. That's not a negative, that's just a bracket. So the input, as we said, is 2 acetyl coenzyme A, and the outputs are 2 ATP, 4 carbon dioxide, 6 NADH, and 2 FADH2. Basically, every single acetyl coenzyme A spins the Krebs cycle. It does one whole rotation, and one whole rotation does all these outputs divided by 2, so it would just be 1 ATP. You get the picture. But all you need to focus on for right now is 2 ATP. Everything else here, this is just, it goes elsewhere in the body. We're not focused on that right now. The ATP from here moves into this, the electron transport chain, which, this also occurs in the mitochondrial matrix, just like this one down here. The arrow, it moves, but it doesn't leave the mitochondrial matrix, but it will here. The ATP is moved to the electron transport chain, which is in the inner membrane of mitochondria. The electron transport chain, it's a collection of proteins. The electrons are falling, they're not physically falling, you can just imagine it. And that makes proteins go from reduced, remember, oil rig oxidated is lost electrons, and then reduced is gained electrons. So they go from reduced, gaining electrons, which means they accept it, to oxidized state, which means they donate it or they lose it. So as you can see, this is the reduced state. It's taking in electrons. E minus is electron. It's taking in electrons. It goes right here, flips upside down into 2, and now it's no longer taking electrons from this area. It's donating it instead, and they're moving over here. This doesn't produce ATP directly, but something really important, you got to know this for sure, is the final electron acceptor is oxygen. This electron is going to meet some oxygen, because this electron will be here, right there. O2 pairs with 2H plus and 2 E minus, which is 2 electrons and 2 positive hydrogen atoms. So the 2 positive and the 2 negative, they cancel out. So now just think of it as O2 pairing with 2H, which you know what that makes, H2O. And, I didn't miss anything. But anyway, The ETC creates a proton gradient, which means that as proteins move in, they pump H plus into space. So if you, I'll show it in a second, but as you're pumping in proteins from the electron transport chain from above the H pluses and they move down, that is powering chemiosmosis. And side note, hydrogen atoms power cell work, but this chemiosmosis that is powered, you gotta move over here. And this right here, chemiosmosis, this is the ATP synthase, which I'll show right now. And the enzyme, it makes ATP from ADP plus P. So if you can see the ADP and the P at the bottom there, that works really well. ADP and P are added, and this makes ATP, the arrow ends here. This uses energy from the H plus, as you can see at the top. It uses it from the H plus gradient, which is what we saw over here in the electron transport chain. And something important that I did miss over here with glucose is that, see this O, since oxygen is 3, O3, it's negative, which is why when I said oil rig and oil rig, it's oxidation, it loses electrons, it's losing then minus to just become like neutral NADH. That's why it's not negative or anything. And then down here, this is your net gain from every stage. So from glycolysis, you gain 2 ATP. Remember, you lose 2, gain 4, your net is 2. Pyruvate oxidation, you don't gain anything here. All you're doing is the coenzyme A is linking to the pyruvate to make acetyl-coenzyme A. Citric acid cycle, also known as the Krebs cycle. Remember, your inputs are 2 acetyl-coenzyme A. Your outputs are 2 ATP. That's why you gain 2 ATP. And then oxidative phosphorylation, which the big word, it literally means the same thing as electron transport chain plus chemiosmosis, which is known as the proton gradient. This is where the money's at. This is where you get 26 to 28 ATP for a total of 30 to 32. The reason that there's variation is because sometimes the plant messes up in here or something gets messed up and energy is lost where it shouldn't be. So I can't really give an example because I don't know, but that's why these vary. And as I said, so quick recap, glycolysis occurs in the cytosol. Lose 2 ATP, gain 4, 2 ATP, that's your net. Pyruvate oxidation occurs in the mitochondrial matrix. Coenzyme A links to the pyruvate, that's why you don't gain anything. Citric acid cycle, Krebs cycle, inputs are the 2 acetyl-coenzyme A from here, also occurs in the mitochondrial matrix. Both of them, those are important to know. These ones, that you take it up here, it makes it 2 ATP, that's why you have 2 ATP as a net. Finally, the electron transport chain plus chemiosmosis, known as oxidative phosphorylation, it's a big word. You should know 26 to 28. And this is all 26 to 28 per glucose. So every time a glucose enters in, these ones, or the oxidative phosphorylation will produce 26 to 28 ATP per glucose. And we add all them up, 30 to 32. So that concludes this very long unit.